Part 1. The Body as a Lipid System. Chapter 1. The Lipid Architecture of the Human Body. The human body is usually explained from the outside inward. Bones create the frame, muscles generate movement, organs carry out specialized tasks, and food supplies the energy required to keep everything running. That sequence is familiar, but it hides the level where most biological behavior is actually decided. Life does not begin at the scale of the visible body. It begins at the scale of boundaries. Every cell survives by building a physical edge between what belongs inside and what remains outside. That edge is not a passive wrapper. It is an active material system made largely from lipids, organized into membranes with specific thickness, fluidity, curvature, charge behavior, and receptor placement. Before the body can think, contract, digest, detoxify, repair, or defend itself, it must first construct stable surfaces that can hold gradients, embed proteins, receive signals, and control exchange. The body is built from lipids in a much more literal sense than most nutrition language allows. Not because fat is merely stored in tissue or burned for fuel, but because lipid structure forms the basic material platform on which cellular function sits. This changes the way anatomy should be understood. A cell is not simply a bag of fluid containing useful parts. It is a compartment made possible by membrane architecture. The mitochondrion does not generate energy in empty space. It depends on highly specialized membranes whose geometry and lipid composition allow electron transport proteins to sit in the correct position, pass electrons in sequence, and pump protons across a controlled barrier. The nucleus does not protect DNA through abstraction. It uses a membrane envelope with selective pores, anchored structural proteins, and tightly managed transport. Synapses, immune cells, endocrine glands, intestinal barriers, hepatocytes, and muscle fibers all depend on lipid-organized boundaries to decide what enters, what leaves, what binds, what activates, and how quickly the system responds once activation begins. The visible body is therefore the large-scale expression of countless microscopic interfaces. If those interfaces are stable, signaling becomes more precise. If they become distorted, oxidized, overly fluid, overly rigid, or compositionally disordered, function becomes noisy before disease becomes obvious. Lipids are uniquely suited to this role because they solve a physical problem that proteins and carbohydrates cannot solve alone. They self-organize into bilayers because one end interacts with water and the other resists it. That amphipathic design creates separation without requiring external scaffolding. From that simple physical property, biological order becomes possible. A membrane can hold an electrochemical gradient because the lipid barrier slows uncontrolled ion movement. A receptor can detect an outside signal and convert it into an intracellular response because the membrane provides both anchoring and local organization. An enzyme can work efficiently because the surrounding lipid environment alters its orientation, mobility, and access to substrates. This is why the chemistry of lipids cannot be reduced to calorie value. A fat molecule is not important only because it can be oxidized for ADP. Its importance begins earlier at the level of arrangement. Which fatty acids are present where they are placed within phospholipids, how tightly they pack, how vulnerable they are to oxidation, and which neighboring molecules sit beside them all influence the behavior of the membrane that cell must live through. Once this is understood, the old division between structure and signaling starts to collapse. In biology, structure is signaling. The membrane is not just the surface on which signaling occurs. It shapes which signals can occur, how strongly they propagate, how long receptors remain clustered, how channels open, how transporters cycle, and how inflammatory enzymes access their substrates. A receptor embedded in a stable, properly organized membrane does not behave the same way as that same receptor embedded in a disordered membrane loaded with damaged lipids or distorted by chronic metabolic stress. The protein may be identical, yet its performance changes because the physical environment around it has changed. The cell does not experience molecules as isolated concepts. It experiences them as material conditions. Thickness, viscosity, lateral mobility, oxidative load, lipid raft integrity, cholesterol distribution, and phospholipid saturation all alter what the cell can do in real time. This makes membrane composition one of the most overlooked determinants of system behavior across the entire body. The term membrane often sounds flat and simplistic, as if it describes a thin film with a uniform job. Real membranes are more complex than that. They are dynamic assemblies containing phospholipids, cholesterol, sphingolipids, glycolipids, embedded proteins, peripheral signaling complexes, and localized microdomains that behave differently from the regions around them. Some parts of the membrane are more ordered and tightly packed. Some are more fluid. Some regions favor receptor clustering. Some favor vesicle formation, endocytosis, exocytosis, or immune synapse formation. The membrane is closer to a living surface than a generic envelope. It sorts activity spatially. It creates zones of probability. It determines whether molecules collide, whether enzymes find their targets, whether signaling stays local or spreads, and whether mechanical stress can be absorbed without tearing the underlying logic of the cell apart. When membrane composition drifts, the body does not merely become deficient in some abstract way. It begins making decisions through altered surfaces. This is especially important in tissues that must signal rapidly or maintain strict discrimination. The nervous system depends on membranes that can support fast electrical behavior, synaptic vesicle cycling, receptor sensitivity, and long-range structural maintenance. The immune system depends on membranes that can detect threat, form immunological synapses, reorganize receptors during activation, and later generate the lipid mediators that help terminate inflammatory activity. The intestinal lining depends on epithelial membranes and junctional structures that preserve barrier function while still permitting selective absorption. The liver depends on membrane-intensive trafficking, packaging, detoxification, and bioproduction. Endocrine tissue depends on membrane receptor fidelity and transport precision. In each case, function emerges from architecture. Cells are not just using lipids in the background. They are behaving through them. The body's internal communication network is therefore inseparable from lipid geography. Hormones can circulate through blood, neurotransmitters can cross synapses, cytokines can move through tissue, but the final effect of every one of those signals still depends on whether the target membrane can receive and translate the message correctly. Signal reception is not simply about ligand availability. It is about receptor conformation, receptor density, receptor mobility, membrane charge landscape, local cholesterol content, and the presence of neighboring lipids that either stabilize or distort the signaling platform. When people talk about receptor resistance, inflammatory sensitivity, or impaired cellular response, they often jump immediately to hormones, genes, or deficiencies. Those matter, but the membrane is where the instruction is either executed cleanly or degraded into noise. A cell can only respond with the precision that its structure allows. Energy production follows the same rule. Mitochondria are often discussed as if they were little power plants floating inside the cell, generating ATP as long as nutrients arrive. That picture is incomplete because mitochondrial output depends on membrane architecture at every stage. The inner mitochondrial membrane holds the electron transport chain, maintains the proton gradient, and organizes the geometry required for oxidative phosphorylation. Cardiolipin and related membrane components help stabilize respiratory complexes and preserve Christie structure. If that membrane becomes compositionally damaged, oxidatively injured, or physically unstable, electron flow becomes less efficient, proton leakage rises, reactive oxygen species can increase, and ADP generation becomes more costly. The problem does not begin only at the level of fuel delivery. It begins at the level of whether the machinery has a coherent surface on which to operate. Biological energy is not produced by nutrients alone. It is produced by nutrients entering well-built structures. Inflammation also has a structural dimension that is often ignored. Immune activation is not simply a matter of white blood cells releasing inflammatory molecules. It is built from membrane events, receptor assembly, arachidonic acid release, eicosanoid synthesis, vesicle trafficking, redox signaling, and later the generation of specialized pro-resolving mediators from omega-3 substrates. That means the inflammatory response inherits the strengths and weaknesses of the membrane systems that support it. A body with damaged lipid architecture may still initiate inflammation aggressively because initiation pathways are easy to trigger, but may struggle to organize resolution once the acute threat has passed. The result is not just excess inflammation in the abstract. It is incomplete inflammatory sequencing. The system enters defense mode, spends resources, damages tissue, and then fails to exit cleanly because the underlying lipid environment does not support proper closure. The architecture of the membrane shapes the timeline of immunity. This is one reason the phrase "fat is fuel" is too narrow to explain human biology. Fat can absolutely be used as fuel, and in many tissues it is an efficient and stable energy source. But if fat is discussed only in metabolic terms, the deeper logic is lost. Lipids are also insulation, signal precursors, barrier material, transport components, organelle-defining structures, and determinants of protein behavior. They influence mechanical resilience, oxidative vulnerability, membrane permeability, and the timing of inflammatory transitions. They participate in bioformation, myelin integrity, steroid hormone synthesis, and intracellular trafficking. When the nutritional conversation reduces fat to a question of caloric density, it strips away the reason lipid quality matters. Calories describe combustion potential. They do not describe membrane suitability. Two diets can deliver similar calories while building very different tissue behavior over time because the body is not made from calories. It is made from matter that must be arranged correctly. The concept of lipid architecture also clarifies why chronic dysfunction often develops gradually and diffusely rather than through one clean failure point. A membrane can become slightly more oxidized, slightly more disordered, slightly less responsive, and slightly less capable of supporting receptor fidelity without producing an immediate dramatic symptom. Early changes appear as noise: slower recovery, less stable energy, mild inflammation, greater reactivity, poorer stress tolerance, altered mood, weaker barrier integrity, or reduced metabolic flexibility. Over time, those small distortions compound across tissues. The nervous system becomes less stable. The immune system becomes less discriminating. The gut becomes more permeable. Mitochondria become less efficient. Endocrine responses become less predictable. None of these failures are fully separate because they are all being expressed through the same class of material surfaces. The body begins to behave like a system whose interfaces have degraded. This chapter sets the foundation for everything that follows because omega-3 only makes sense within this structural frame. If the reader approaches omega-3 as a supplement that does things, the discussion stays shallow and eventually becomes contradictory. If the reader understands that omega-3 enters phospholipids, alters membrane behavior, changes substrate availability for inflammatory and pro-resolving mediators, and gradually modifies how cells organize signaling, then the subject becomes coherent. Omega-3 is not important because it produces a quick benefit. It is important because it changes the material conditions through which benefits become possible. That is a slower idea, but it is the more biologically honest one. The human body is therefore not best understood as a calorie processing machine with occasional structural needs. It is a structure-dependent system that uses energy to maintain organized matter. First, the boundary has to hold. Then gradients can exist. Then proteins can work in place. Then signaling can become specific. Then tissues can coordinate. Then the organism can behave coherently across time. Lipids sit near the beginning of that chain. They are part of what makes a cell, an organelle, and a tissue capable of organized function rather than biochemical drift. Once that is seen clearly, every later question about inflammation, metabolism, brain health, cardiovascular disease, immune regulation, and omega-3 biology can be asked at the right level. The body is built from lipids. The body is commonly described as if its most important materials are obvious to the eye. Bone is treated as the frame, muscle as the engine, organs as the machinery, and food as the fuel supply that keeps the machinery running. That picture is not wrong, but it begins too late in the hierarchy. Before any organ can perform a task, before any tissue can specialize, and before any cell can maintain identity, the body has to solve a more primitive problem. It has to create boundaries that are stable enough to hold chemistry in one place, selective enough to control exchange, and flexible enough to change behavior without losing integrity. Those boundaries are built largely from lipids. Every cell depends on a membrane. Every organelle depends on a membrane. The body is not simply surrounded by fat, nor does it merely store fat for later use. It is assembled through lipid-based surfaces that determine where one biological space ends and another begins. Without those surfaces, there's no controlled metabolism, no electrical gradient, no receptor signaling, no mitochondrial output, no digestion, no immune discrimination, and no nervous system. There is only chemical mixing. Lipids matter because they create organized separation in an aqueous environment. Water dominates the chemistry of the body, and that fact makes membrane formation necessary. A living system cannot function if all molecules move freely without compartmental control. The cell needs an inside that is chemically different from the outside. The mitochondrion needs an inner membrane that can hold a proton gradient. The nucleus needs an envelope that protects genetic material while still allowing controlled communication. Vesicles need lipid surfaces so cargo can be moved without being dumped into the wrong compartment. The intestine needs epithelial membranes that can absorb selected materials while refusing others. The nervous system needs myelin-rich lipid insulation and membrane surfaces capable of rapid electrical change. In each case, lipids are not background filler. They are the material solution that allows biological order to exist at all. The body does not first become alive and then add lipids as supporting accessories. It becomes organized because lipids make organized biological space physically possible. This is why the usual nutritional framing of fat is too small. Fat is often reduced to energy density, body composition, appetite, or storage. Those categories are real, but they do not reach the deepest function of lipids in human physiology. A lipid is not important only because it can be oxidized. It is important because it can become part of a membrane, alter the packing of neighboring molecules, change permeability, modify receptor mobility, influence enzyme access, and serve as the stored substrate for later signaling molecules. A body built from unstable or poorly balanced lipids does not merely carry a bad nutrient profile on paper. It behaves differently. Receptors cluster differently. Inflammatory triggers spread differently. Immune cells activate and resolve differently. Mitochondria hold gradients with different efficiency. Barriers leak more easily or become harder and less adaptive. The quality of the material changes the behavior of the structure, and the behavior of the structure changes the behavior of the organism. At the cellular level, this means function always sits on top of composition. Two cells can contain the same genes, the same proteins, and the same broad purpose, yet behave differently because their membranes are built differently. The physical environment surrounding membrane proteins changes how those proteins move, how they interact, and how reliably they transmit information. A receptor is not floating in abstraction. It is embedded in lipid matter. Its activity is shaped by membrane thickness, fluidity, saturation, cholesterol distribution, local curvature, and oxidative stability. Ion channels, transporters, docking machinery, inflammatory enzymes, and signal complexes all inherit the conditions of the membrane that holds them. When people think about health in terms of hormones, neurotransmitters, or immune messengers alone, they often miss the layer where those messages are actually interpreted. The cell reads the world through material surfaces. Change the surface, and you change the reading. This structure-first perspective also explains why lipid biology reaches into every major system at once. The brain is enriched with specialized lipids because rapid signaling, synaptic plasticity, membrane excitability, and long-lived neural stability all require carefully built surfaces. The immune system depends on membrane reorganization to detect threat, build inflammatory synapses, release signaling molecules, and later transition toward resolution. The cardiovascular system depends on endothelial membrane behavior, lipoprotein transport, and vascular signaling that all emerge from lipid-organized interfaces. The liver packages lipids, modifies them, exports them, and uses membrane-intensive trafficking systems to manage the body's chemical load. The gut maintains a boundary with the external world through an epithelial surface whose composition influences permeability, immune tone, and nutrient handling. These are not separate stories. They are variations of the same underlying rule. Life is managed through boundaries, and biological boundaries are lipid structures under constant metabolic maintenance. Once that is understood, the phrase "the body is built from lipids" becomes literal rather than metaphorical. It does not mean the body is made only of fat, and it does not mean every health problem reduces to one nutrient class. It means that lipids occupy a foundational position in biological organization because they help determine the architecture through which everything else operates. Proteins do work, but much of that work depends on being placed into the right membrane context. Genes encode instructions, but those instructions still have to be executed inside compartments defined by lipid surfaces. Energy can be generated from many substrates, but efficient energy production still depends on membranes that can hold gradients and organize electron flow. Even inflammation, which is often described as a purely signaling phenomenon, depends on lipid-derived mediator pathways and membrane events that decide how strongly a response begins and whether it can end cleanly. This is the level at which omega-3 and the broader lipid conversation have to be approached. The question is not simply whether a given fat has benefits. The deeper question is what kind of structure that fat helps build over time. Does it strengthen membrane performance or weaken it? Does it improve signaling fidelity or increase instability? Does it support resolution chemistry or only add more susceptibility to damage if the surrounding environment cannot protect it? Does it enter a body already saturated with competing inflammatory substrates and oxidative stress? Or does it become part of coordinated reconstruction of membrane architecture? Those are structural questions, and they matter because the body expresses its health through the materials from which it is built. Long before symptoms become visible, the architecture has already been changing. Membranes as the foundation of biology. Biology begins when matter is separated into controlled spaces. That separation is not philosophical, and it is not optional. Without it, there is no cell, no organelle, no metabolism, no signaling sequence, and no stable life process that can persist long enough to become tissue, organ, or organism. The membrane is the structure that makes this possible. It creates an inside and an outside, but more importantly, it allows those two spaces to remain chemically different while still permitting selective exchange between them. That is the first real act of biological organization. A membrane holds gradients, localizes reactions, embeds proteins, and gives the cell a stable surface through which information can be received and acted on. Once that surface exists, the rest of biology can begin layering itself on top of it. Without the membrane, there is no meaningful distinction between environment and organism. There is only chemistry dispersing into chemistry. This is why membranes should not be treated as passive wrappers or thin packaging around more important internal machinery. The membrane is part of the machinery. In many cases, it is the condition that allows machinery to function at all. Ion channels require a membrane, so ions can move across a meaningful barrier rather than through open fluid. Receptors require a membrane, so outside information can be converted into intracellular action. Transporters require a membrane so nutrients, waste, and signaling molecules can be selectively moved rather than randomly diffused. Enzymes associated with the membrane often behave differently depending on the exact lipid environments surrounding them. A protein does not simply sit in a membrane like a bolt fixed into plastic. It exists in a material field that changes its position, motion, conformation, binding behavior, and access to substrates. The membrane is therefore not just where proteins are located. It is part of what determines what those proteins can actually do. At the scale of the whole cell, membrane structure defines identity. A cell does not remain alive because it contains useful molecules. It remains alive because those molecules are organized within a bounded system that can maintain concentration differences, electrical gradients, pH differences, osmotic balance, and controlled traffic. The cell membrane prevents collapse into equilibrium. That matters because equilibrium in a living system is often death. Life requires maintained asymmetry. Sodium has to be kept in one distribution, potassium in another, calcium must remain tightly controlled, and metabolites must be directed rather than allowed to drift. The membrane is what gives the cell leverage over its own chemistry. It allows the cell to resist the passive tendencies of the surrounding environment and hold a state that is energetically costly but biologically useful. In that sense, the membrane is not merely a boundary. It is a tool for preserving non-randomness. The same principle repeats inside the cell. The mitochondrion is not just a structure floating in cytoplasm. It is a membrane-built energy system whose inner membrane holds the electron transport chain and creates the proton gradient required for ADP production. The nucleus uses a membrane envelope to protect genetic material and control traffic between the genome and the rest of the cell. Lysosomes, peroxisomes, endosomes, secretory vesicles, and the endoplasmic reticulum all depend on membranes because each one performs chemistry that must be spatially separated from other chemistry. Once this is seen clearly, it becomes obvious that membranes are not a feature of biology. They are one of its core organizing laws. Life works by building compartments, and compartments only function when membranes are stable enough to preserve distinction while dynamic enough to support exchange and adaptation. This turns membrane composition into a major determinant of physiology rather than a minor biochemical detail. A membrane is not interchangeable with any other membrane. Its behavior depends on which phospholipids are present, which fatty acids occupy those phospholipids, how much cholesterol is present, how sphingolipids are distributed, how ordered or disordered the surface becomes, and how vulnerable the structure is to oxidation or mechanical disruption. Those factors shape permeability, thickness, flexibility, curvature, electrical behavior, receptor clustering, and signaling fidelity. A membrane that is too rigid creates one kind of problem. A membrane that is too unstable or oxidatively damaged creates another. A membrane overloaded with poorly protected polyunsaturated material may allow high signaling flexibility while also becoming chemically fragile. A membrane built with greater structural stability may preserve gradients and protein behavior more reliably but alter mobility in other ways. Biology is constantly solving this material balancing act. The health of the system depends on whether that balance remains coherent across tissues and over time. That is why membranes sit underneath so many processes that are usually discussed separately. Neural firing depends on membrane excitability and ion channel behavior. Hormone signaling depends on receptor function within specific membrane environments. Immune activation depends on membrane reorganization, receptor assembly, vesicle trafficking, and later the release of lipid-derived mediators. Barrier tissues depend on membrane integrity plus the protein structures anchored to them. Mitochondrial output depends on the ability of membranes to maintain proton separation and organize respiratory complexes. Even mechanical behavior at the tissue level depends partly on whether cell membranes and associated lipid structures can absorb stress without destabilizing the signaling systems built into them. The body looks like many systems from the outside, but at depth those systems are repeatedly operating through variations of the same membrane logic. This also explains why biological breakdown often begins long before conventional disease categories become obvious. Membranes can lose quality gradually. They can become more oxidized, less stable, more compositionally distorted, less able to support receptor precision, less effective at holding gradients, and more likely to amplify inflammatory noise. The early signs may appear disconnected. A person notices unstable energy, poorer recovery, more reactivity to food or stress, changes in mood, reduced exercise tolerance, worsening metabolic control, or a greater tendency toward chronic irritation in tissues. These do not always look like membrane problems because the membrane is not visible in ordinary medical language. But much of what the person is experiencing may be the downstream behavior of surfaces that no longer organize biology cleanly. The system has not lost all function. It has lost structural precision. Seen from this angle, the membrane is where structure and signaling become inseparable. It is the place where matter becomes information-bearing. A receptor does not just detect a hormone because the hormone exists. It detects it in a certain membrane state, with a certain lipid neighborhood, under a certain curvature and cholesterol distribution, with certain probabilities of clustering and internalization. An inflammatory enzyme does not merely act because the gene for that enzyme is expressed. It acts because the membrane provides the substrate access and physical conditions needed for action. A mitochondrion does not merely burn fuel because fuel is present. It produces energy because membrane architecture permits organized electron flow. Function is therefore not suspended above structure. Function is the behavior of structure under conditions. For that reason, any serious discussion of health has to begin lower than symptoms, lower than blood markers, and lower than even many familiar biochemical pathways. It has to begin at the level of the material surfaces that make organized life possible. Membranes are the foundation because they are the condition for compartmentalization, signaling, energy conversion, selective exchange, and biological identity itself. Once that is understood, the rest of physiology starts to align. Nutrients stop looking like isolated chemicals and start looking like materials that alter surfaces. Chronic disease stops looking like a list of unrelated malfunctions and starts looking more like the accumulated consequences of structural distortion. And omega-3, which is so often discussed as a supplement with benefits, can finally be placed where it belongs, inside the membrane story, where biology actually begins. Compartmentalization and control. Life depends on separation. Not symbolic separation, but physical separation that allows different chemical conditions to exist at the same time inside the same organism. A living body cannot function as one open fluid space where everything mixes freely. If that happened, gradients would collapse, reactions would interfere with one another, signaling would lose direction, and the distinction between useful chemistry and destructive chemistry would disappear. Compartmentalization is the solution to that problem. It allows the body to divide itself into controlled zones, each with its own composition, pressure, charge distribution, substrate concentration, and functional purpose. The membrane is what makes that division real. Once a membrane defines a compartment, biology gains the ability to control what enters, what leaves, what accumulates, what reacts, and what remains excluded. That is the beginning of regulation in the most literal sense. Control is not first a matter of hormones, genes, or feedback loops. It is first a matter of whether the system can hold separate spaces long enough for those higher forms of control to even exist. The cell itself is the first major example. It maintains an internal environment that is chemically different from the fluid surrounding it, and that difference is not incidental. Intracellular potassium remains high while sodium is kept relatively low. Calcium is held under tight restriction because even small increases can trigger powerful downstream effects. pH is buffered within narrow ranges because enzyme systems depend on local chemical stability. Nutrients are brought in selectively, waste is moved out selectively, and signaling molecules are either admitted, excluded, degraded, recycled, or transported according to need. None of this happens in an open system. It depends on membranes that can preserve concentration differences against the constant tendency of molecules to diffuse toward equilibrium. In practical terms, the membrane allows the cell to say no. It can refuse indiscriminate entry. It can prevent the loss of critical molecules. It can control timing. It can turn transport into an active decision rather than a passive leak. Inside the cell, the same logic becomes more elaborate. The body does not place all chemistry into one intracellular pool and hope that specificity will emerge from molecular goodwill. It builds internal compartments because precision requires separation. The nucleus isolates the genome from the cytoplasmic environment while still allowing tightly regulated communication through nuclear pores. The mitochondrion creates two membrane-defined spaces so electron transport can build a proton gradient across the inner membrane and convert that gradient into ATP. Lysosomes contain acidic degradative chemistry that would be destructive if released without control. Peroxisomes handle oxidative reactions that need their own boundary conditions. The endoplasmic reticulum organizes protein synthesis, folding, and lipid handling within its own membrane network. Vesicles move cargo in enclosed form because transport without containment would create chemical disorder. Compartmentalization, therefore, does not just make biology tidy. It prevents incompatibility. It allows one reaction to occur intensely in one place without corrupting the conditions needed somewhere else. This creates a deeper definition of control than the word usually carries. In ordinary language, control sounds like command from above, as if the body is centrally managed by regulatory molecules issuing instructions down a chain. Biology does use layered signaling, but that signaling only works because compartments constrain possibility. A hormone may circulate systemically, but its effect still depends on whether the target tissue can receive it, buffer it, amplify it, or terminate it within the architecture of its compartments. A calcium signal can activate contraction, secretion, or transcription depending on where calcium rises, how fast it rises, how long it remains elevated, and which local proteins are present to interpret it. A reactive oxygen species can serve as a controlled signal in one compartment and become damaging chemistry in another. Control is therefore never just about the presence of a molecule. It is about where that molecule exists, where it is prevented from existing, and how the surrounding boundary structure limits its reach. This is why loss of compartmental integrity produces disproportionate consequences. When a barrier fails, chemistry that was supposed to remain local spreads into spaces that were not built to handle it. When mitochondrial membranes lose integrity, proton gradients weaken, ADP production becomes less efficient, and electron leakage can rise. When lysosomal contents escape, degradative processes begin damaging the wrong targets. When intestinal barrier function deteriorates, microbial fragments, incompletely processed food components, and inflammatory stimuli gain greater access to immune surfaces beneath the epithelial layer. When vascular or tissue barriers become more permeable, fluid shifts, immune traffic, and signaling noise increase. These are not merely examples of damage occurring in isolated places. They are examples of lost control through failed boundaries. Once compartmentalization weakens, the system loses the ability to keep processes in their proper zones, and biological behavior becomes less selective, more expensive, and more chaotic. The nervous system offers one of the clearest demonstrations of this principle. Neurons rely on membrane-defined compartments to maintain resting potential, generate action potentials, release neurotransmitters at synapses, and recover after signaling events. The signal only works because ions are separated, channels open in a controlled sequence, and neurotransmitter release occurs into a narrow synaptic space rather than into general tissue fluid. The brain as a whole depends on larger compartmental controls as well, including the blood-brain barrier, specialized extracellular environments, and glial regulation of local chemistry. Remove those layers of separation and neural precision degrades quickly. The same is true in the immune system, where discrimination depends on controlling where antigens are encountered, where inflammatory mediators are generated, where immune cells traffic, and when damaged material is cleared without spilling its contents across surrounding tissue. The body maintains intelligence through boundaries. Without boundaries, information turns into noise. Lipid structure sits underneath all of this because membranes are the physical basis of compartmentalization. Their composition determines how effectively a boundary can hold gradients, resist uncontrolled permeability, support transport proteins, recover from stress, and preserve signaling precision under load. A membrane that is compositionally weak, excessively oxidized, overly permeable, or mechanically unstable may still exist, but does not provide the same level of control. It allows more leakage, more misfiring, more unintended molecular contact, and more distortion of timing. A stable membrane does the opposite. It sharpens distinction between compartments. It supports selective transport instead of indiscriminate movement. It preserves the conditions needed for high-resolution biological behavior. This is one reason lipid quality matters so much beyond the crude language of energy intake. The body is not just fed by fats. It is governed through structures partly made from them. Compartmentalization also explains why chronic dysfunction often feels diffuse before it becomes diagnosable. Control rarely disappears all at once. More often it erodes gradually. Barriers become slightly more permissive. Membranes become slightly less reliable. Signaling molecules persist a little longer in places where they should have been cleared. Oxidative reactions reach a little further than they should. Immune recognition becomes slightly less selective. Energy production becomes slightly less contained. Early on, this does not look like one dramatic pathology. It looks like accumulated imprecision. The person becomes less resilient, less stable, less capable of clean transitions between rest and stress, activation and recovery, feeding and fasting, challenge and repair. The compartments are still present, but the quality of control they provide has declined. Seen clearly, compartmentalization is not a side feature of biology. It is what allows control to exist in the first place. The body does not regulate by floating commands through undifferentiated chemistry. It regulates by building spaces, protecting those spaces, and using those spaces to determine what kind of reactions are possible and where. Once that framework is established, signaling becomes meaningful, energy conversion becomes efficient, immune defense becomes discriminating, and tissue behavior becomes coordinated instead of random. This is why membrane integrity and lipid architecture deserve such central attention. They are not one topic among many. They are part of the physical basis by which the body holds order against collapse. Lipids as structure, not just fuel. One of the most limiting ideas in modern nutrition is the habit of treating fat primarily as stored energy. In that framework, lipids matter because they contain calories, slow digestion, influence appetite, and can be burned when glucose is not immediately available. All of that is true, but it describes only one layer of lipid biology and not the deepest one. The more foundational role of lipids is structural. They are part of the material from which the body builds itself. They form membranes, define compartment boundaries, shape organelle behavior, influence tissue stability, and create the physical environment in which proteins, receptors, enzymes, and transport systems must operate. When lipids are viewed only as fuel, the body gets interpreted like an engine that occasionally uses fat for combustion. When lipids are viewed structurally, the body looks different. It becomes a built system whose performance depends on the quality, balance, and durability of the materials used to construct its surfaces. This matters because the body does not merely use lipids after they are eaten. It incorporates them. Fatty acids are esterified into phospholipids, packed into membranes, stored in droplets, transported in lipoproteins, converted into signaling molecules, and positioned within tissues according to ongoing metabolic need. Once incorporated, they change the behavior of the structure they enter. A membrane containing different lipid species does not just look different on a chemistry chart. It behaves differently under stress, responds differently to signals, and supports different patterns of receptor movement, ion transfer, permeability, and enzyme activity. That means dietary fat is not simply entering a furnace. It is contributing to the construction, repair, and remodeling of biological matter. This is one reason why the same calorie intake can produce very different physiological outcomes depending on what kinds of lipids are repeatedly being supplied and what broader metabolic conditions those lipids enter. The structural role of lipids begins with the membrane, but it does not stop there. Lipids determine how tightly a membrane packs, how much lateral movement occurs within it, how vulnerable it is to oxidation, how easily it bends to form vesicles, how stable it remains under mechanical stress, and how effectively it can host proteins that need a certain physical environment in order to function well. Cholesterol contributes to membrane order and local stiffness. Saturated fatty acids tend to support tighter packing and greater structural stability. Monounsaturated fats can support flexibility while maintaining relative resilience. Polyunsaturated fats increase fluidity and expand signaling potential, but they also introduce greater chemical fragility because their double bonds are more susceptible to oxidation. None of these characteristics are abstract. They alter how real tissues behave. A neuron, an immune cell, an intestinal epithelial cell, and a hepatocyte all inherit part of their operating behavior from the lipid composition of the surfaces through which they function. This changes the way energy itself should be understood. The body certainly oxidizes lipids for ATP production, especially in tissues adapted for high rates of fat use, such as skeletal muscle, cardiac muscle, and liver under appropriate conditions. But even here, structure remains upstream. Fat cannot be converted into energy efficiently unless mitochondria are physically capable of doing the work. That requires intact mitochondrial membranes, preserved Christie architecture, controlled proton impermeability, and lipid environments that stabilize respiratory complexes rather than destabilize them. The body does not extract energy from fat in a structural vacuum. It extracts energy through lipid-built organelles whose own function depends on lipid integrity. In other words, lipids are involved on both sides of the equation. They serve as potential fuel, but they also help determine whether the machinery that burns fuel can operate cleanly. The old picture of fat as input and mitochondria as passive burners misses that the machinery itself is partly made from lipid-dependent architecture. The immune system reveals the same principle in another form. Lipids do not only sit in immune cell membranes as inert background material. They help organize receptor clusters, shape the immunological synapse, influence membrane trafficking during activation, and provide the substrate for inflammatory and pro-resolving mediator synthesis. Arachidonic acid, EPA, and DHA are not just nutrients floating in plasma with separate brand identities. They are structural components that can later be mobilized into signaling cascades when phospholipase activity releases them from membrane phospholipids. This means that what appears during inflammation is partly determined by what has already been built into tissue. The inflammatory output of the body is not generated from nothing at the moment of stress. It emerges from stored material. The membrane is acting as both structure and reservoir. That is why long-term lipid intake shapes immune behavior over time, even when no obvious acute symptom is present. The nervous system depends especially heavily on this structure-first view. Neural tissue is dense with lipid material because signal transmission, synaptic vesicle release, membrane excitability, and insulation all require surfaces with specific physical properties. Myelin is lipid-rich because rapid conduction depends on electrical separation. Synaptic membranes require precise composition because neurotransmission depends on curvature, vesicle fusion, receptor organization, and rapid membrane recycling. Brain lipids are not there as emergency calories stored near neurons for convenience. They are part of what allows neurons to behave like neurons. When lipid composition shifts unfavorably, the effect is not limited to energy supply. It can alter signaling stability, membrane excitability, inflammatory sensitivity, and the quality of neural communication itself. That is why the conversation around lipids and brain health becomes shallow when it is reduced to whether a fat feeds the brain. The brain is partly built from lipid architecture, and its behavior follows the quality of that architecture. This structural lens also clarifies why some fats can support stability, while others can increase volatility when the system is not properly protected. A highly unsaturated lipid may contribute useful fluidity and signaling potential, but if oxidative defense is poor, if membrane repair is inadequate, or if inflammatory pressure is already high, that same lipid can become part of an unstable surface that is easily damaged and then converted into reactive byproducts. On the other side, more saturated structures may improve durability and lower susceptibility to peroxidation, but excessive rigidity in the wrong context can also impair certain forms of membrane behavior. Biology is never solved by slogans. The real question is what kind of structure a given lipid pattern creates across a real tissue under real metabolic conditions. This is why the phrase healthy fats often fails to explain anything meaningful. Lipid value depends on role, proportion, tissue context, oxidative environment, and the structural state of the organism already receiving it. Once lipids are understood as structure, the timeline of nutritional change also becomes easier to interpret. Structural remodeling is slower than pharmacologic stimulation. A membrane does not instantly transform because one meal contained more omega-3 or less seed oil. Tissue composition changes over repeated exposure, selective incorporation, turnover, oxidation, repair, and replacement. That is one reason people often misunderstand fat-based interventions. They expect a signaling molecule result from what is actually a remodeling process. Structural inputs work by changing the material conditions of the system over time. Membrane composition gradually shifts. Receptor behavior gradually shifts with it. Inflammatory substrate availability gradually shifts. Mitochondrial resilience, barrier behavior, and signal fidelity can then begin to move in new directions. This is not the pace of a stimulant or a pain reliever. It is the pace of construction. For that reason, lipids should be thought of less as optional dietary macros and more as ongoing building inputs that happen to carry energy. That order matters. Their caloric value is real, but it is secondary to the deeper fact that they enter tissues and influence what those tissues are physically like. The body is not a furnace wrapped in flesh. It is a layered, compartmentalized structure whose surfaces determine much of its behavior. Lipids participate directly in that construction. They help define whether membranes are stable or fragile, whether signals are clean or distorted, whether inflammation can resolve or only persist, whether mitochondria can hold gradients, and whether tissues can preserve order under stress. Once that becomes the frame, the discussion of omega-3, omega-6 saturated fat, phospholipids, cholesterol, and membrane remodeling stops being a side conversation about dietary preference. It becomes a conversation about the materials from which biological performance is built.